All right. New week, week 9. It's exam week, right? So exam on Friday. Meet here. I'll try to get here as early as I can. As soon as I get here, I'll start passing it out. So goal is to get here 10 minutes before 8. Start, and then when you're done, you turn it in. We have that 50 minutes to take it, so you should study. Format will be the same, where you'll have probably a couple true false, and then multiple answer questions, and then you'll have your definitions, which would be for matching. I give you the word bank, I give you the definition, you write the word to match the definition, and then you may have some calculation. And if you think about the calculations that we did, they were really just the calculations from the lab. So population estimation, marker capture lab, estimating population size, or possibly doing some of the biodiversity stuff. Equations would be posted, so you don't have to memorize them. But you do need to know how to use them. So make sure you review that. Our exam goes up until this population. So start reviewing, start studying. All right, let's try that again. How's the sound? All right. Unplug it, plug it back in. That seems to work. All right, so yeah, exam on Friday. Quizzes. If you did not do the quizzes, lecture quizzes, or if you still have a couple attempts left, you have to get it done by noon. They close on noon, they disappear, but at the same time, at noon, that's when the keys go live. So if you took it and you took them on time, you should be able to see the key. I have to go in there and pull down the grades for anyone that took it late. I've got to get them marked. I've got to get them marked, and then once I do get them marked, it'll be good to go. So I plan to get that done this afternoon. So sometime by this evening, everyone should be able to access the key. How do we get to it? Click on your grades, click on the quiz, and then click on the score. Click on the score, it'll open up all the questions, all the answers. It'll show you how you answered and what the correct and wrong answers were. If you don't know how, email me. Any questions? All right, this is where we left off. Population descriptors. Now, population size or density, that was number 1. Spatial dispersion is number 2, and that's this week's lab. We're going outside for it. This week and next week's lab. So there are 3 different patterns of dispersion. There's clumped or aggregated, there's a uniform or regular, and there's a random dispersion pattern. Now when we talk about dispersion, we can calculate a coefficient of dispersion, which is a variance to mean ratio. If we have a variance to mean ratio equal to 1, or approximately equal to 1, that suggests a random pattern. If it's not equal to 1, then it's either going to be uniform or clumped or aggregated. If it's greater than 1, we're the clumped or aggregated. If it's less than 1, it's uniform or regular. And what we talked about was the requirements or the factors that would promote this. So clumped or aggregated is the most common type of pattern that's out there. It can be various factors, including transmission or dispersal stages. So how do the organisms move away from their natal area? Could be due to habitat heterogeneity. Individuals tend to clump towards the better habitats. Could be social patterns or reproductive behaviors, or it could be weather or environmental conditions. All of those things could produce this clumped or aggregated pattern. Random distribution requires a uniform environment that individuals can live everywhere, and it requires a neutral intraspecific interaction. So we can't have either individuals attracting others or repelling other individuals. This random dispersion pattern isn't necessarily common, but it's there. We can see it. There are certain organisms that exhibit this random pattern. And then the uniform regular pattern also has factors that favor its formation. We do benefit from having strong intraspecific interactions, so negative interactions. If one individual's there, they tend to repel other individuals. And also dispersal stages of some plants can produce this type of pattern. So that's where we're going, is this dispersal stages. Because usually the dispersal stages of the plant, those seeds, generate this clumped or aggregated pattern because the seeds tend to drop and they stay near the parent plant. So then when they germinate, they're near the parent plant, and then you just have closely related individuals growing in clumps. But in some cases, you have these seeds that provide a food source for other organisms. And it's these other organisms, their behavior, their foraging, that tends to drive the formation of the random or the uniform pattern. So this is called the Jansen-Connell hypothesis. Their hypothesis aims at explaining why some of these plants exhibit a random or uniform dispersion pattern. Because again, typically you would think these stages, these seeds, should produce a clumped pattern, but they don't. So their hypothesis says that this uniform or random dispersion pattern relies on seed predators and herbivores. Right, so how does it work? These graphs try to summarize what is going on. All right? So in a normal situation, you get a graph that looks something like this. So you have the number of individuals on the y-axis, and you have distance from the parent tree on the x-axis. So right here at 0, that's the parent tree. And we're going to use, let's say, acorns as our example. Parent plant drops acorns. You're going to have a large number of acorns and a large number of seedlings that'll germinate right near that parent plant, because that's where most of the acorns are. As we go away from our parent plant, we see fewer and fewer individuals, because it's now harder for those acorns to get a larger distance away. Right. Even at the very far ones, they might get there, might hit a rock. You might have an animal that just carries it a little bit farther. Right. Maybe it's part wind. Helps to push it away from the parent plant. But you can see most of the acorns, most of the seedlings, the young seedlings, are occurring right near our parent plant. All right. If we account for seed predation, that tends to change. And seed predation is actually kind of a consequence of the limited dispersal of those acorns, because the large number of acorns below the tree is just like a huge buffet for our seed predator. Right? The tree itself is attracting the seed predators. They're coming. They're going to be much more focused on those areas that have a large number of acorns, and less focused on these that are farther away from the parent plant. So what we see then, after we account for seed predation, is that a lot of these acorns and a lot of these seedlings that are close to the parent plant get consumed. Predator's there, it finds the acorns, it starts eating, and it continues looking in that area for the acorns until basically, if they spend too much time looking for an acorn without finding one, that they say, okay, I'm leaving. They spend their time right around that parent plant. So they clear out all of those plants that are close to the parent tree, and then as you increase your distance, these are the ones that are more likely to escape our predation. Because they're isolated, they're far from the parent plant. You don't have a bunch of other acorns close by attracting those seed predators or those herbivores that are eating the young seedlings. All right, so you've had the attraction of these seed predators and herbivores. They focus on areas where the food is abundant, and they seem to miss those seeds that are farther away from the parent plant. So these guys tend to escape predation. That means then that you're more likely to have these more distant trees forming and growing than the trees that are close together. And it's these guys that start to produce your spacing. They're either the uniform or the random type of spacing across our landscape. So that's our Jansen-Connell hypothesis. Questions? We see it. We've got examples. Acorns, oak trees probably aren't the best. We see it down in the Amazon. A lot of these trees where you do have the seed predators, the herbivores down there, where you do get certain species of trees give this uniform or regular pattern, whereas other types of plants are giving this clumped or aggregated type pattern. All right, questions? All right, so quick summary of this part. So we've defined our populations. We define them based on their demographic discontinuities. All right, so these are the differences in their descriptors. Could be differences in densities, could be differences in their, maybe their dispersion pattern, differences in demographic characteristics, which we'll get to. All right, key point for our populations on how we define populations is that we need all individuals to interact. We have to have the possibility of all individuals interacting. Doesn't necessarily need to randomly mate, but they need to interact. All right, if you have your boundaries, your limits set up so that all individuals interact, or not just interact, they don't just interact, but they randomly mate, then that defines our deme, which evolution acts on. All right, so the ecological versus evolutionary population, that's kind of that distinction. Ecological interaction, they all have to interact, they don't have to necessarily randomly mate. Evolutionary population, they have to randomly mate, and thus by definition they are interacting. All right, we do have the three fundamental descriptors of our populations. We started with the first two, population size and density, and then dispersion is that second one. So population size and density, crude versus ecological density. Probably ecological density is a little bit more useful. That's probably going to be the more likely one that you see. However, crude density is a quick and dirty way where you don't have to account for appropriate habitat. And we just finished up dispersion, the three different types. Talked about Jansen-Connell hypothesis just now. That's a quick summary of our size and dispersion. Questions? All right, if not, we will move on. So I do have a quiz set for the demographics. I think what we will do is I'll get it up, but the due date will be probably next week. We'll give you this week to kind of study for the exam, and then the due date for the population size dispersion probably maybe on Tuesday. Probably on Tuesday night. So It'll be after the due date, after our exam. It'll be after the exam. All right, third set descriptor of our populations. It's the demographic characteristics. All right, demographic characteristics. So we've got our three fundamental descriptors. Demographic characteristics will include our age structure, sex ratios, and birth rates. That's what we're going to address. We're going to focus on age structure, sex ratios, and birth rates. And the birth rate would be the number of offspring that you have. Do you have a lot of offspring? Do you have a few offspring? And these aren't necessarily independent of each other. Oftentimes the birth rates depend on the age structure. Right, sex ratios can change with the age structure. It's these things that we're going to focus on, we're going to talk about. Ready? Head shakes. While you're finishing up, Tri-Beta meeting, 20th. It's here, Texas Tech's Biomedical PhD program, if you're interested. And if you're interested in talks, we have a teaching demonstration in here at 12 o'clock. Botany professor candidate, and then research talk is at 3. They say they start at like 12:15 or 3:15. But they're in here if you're interested. All right, we ready? All right, demographic characteristics. Why bother? Well, we look at these because some of these characteristics allow us to predict if a population is going to grow or if the population will decline. Now, it's not a guarantee, it's a prediction. But usually the prediction is pretty decent. We know the age structure, if we know the sex ratios, if we know the birth rates. One or combination of these things tells us, yeah, this population looks like it's growing. It's probably what we would call a healthy population because it can grow. Other situations, based on the structure, based on the ratios, it looks like what we would call an unhealthy population because the population could be in decline. So what are each of these? Well, age structure is how many individuals are there in each of the various age groups or age classes. All right. If we see a large number of individuals in our population that are close to approaching reproductive maturity, tends to suggest we're getting ready to see a lot of reproduction occurring, a lot of offspring developing, which probably suggests population growth. If we have a large population or a large number of individuals in our population that are at the tail end of their reproductive age, the prime reproductive age, so that they're basically getting past that mark, that's probably a sign that our population will start to enter a decline. You hear this as the aging human population, where they talk about this age structure. Certain developed countries are exhibiting that, where, yeah, just in their population would decline because you've got many more individuals in the population past the reproductive age, but they're not really replacing themselves anymore. Sex ratios play a role because we're talking about the number of females to the number of males, or vice versa. So the most important sex for population growth is males or females? Trick question. They're both needed. But really, in terms of population growth, if we're skewed more towards females, you tend to see populations that increase. Right? If you're skewed more towards male, well, males can't produce have their own offspring, so you tend to see population decline. And then birth rate. How many offspring are born? If you can have a lot of offspring, it might indicate that you're going to have population growth. If you don't have a large number of offspring, maybe you'll see decline. And then also, does this vary by age of the female? So we kind of have to look at this age structure. And just because we have a large number in the reproductive period, they could be at that part where the number of offspring, their birth rates, are actually pretty low. And thus, we're going to exhibit decline. So we can kind of rationalize or make predictions based on these without really knowing or following a population through the years. We can just make predictions. Yeah, we'll increase, or yeah, we'll decrease. All right. So age structure is what we're going to start with. Age structure is the distribution of individuals according to their ages. All right, so we take your population size, and you break those individuals down into different ages or age classes. Or age classes. This age structure is usually presented as a life table. Usually presented as a life table. What is a life table? A life table is a table that shows the age structure and reproductive schedules. All right, so the different types of tables that we have are either a cohort life table or a static life table. A cohort life table is a table where we follow a cohort from birth to death. So what is a cohort? A cohort is those groups of individuals that are all born within about the same time. So the easiest way to explain it would be using an example of annual plants. So the annual plants set their seed and then they die. All right? Our new cohort will be all of those seeds that germinate the next year. So we follow all of those plants that germinated from germination until they die. How do we do it with like humans that have more than one, or they live more than 1 year, and we have overlapping generations? Well, then what we can do is define a cohort based on a specific time period. So we can say all the individuals that were born in 2020, that represents a cohort. And then we follow all of those individuals from when they were born until they die on a yearly basis. Maybe we can do it by month. All individuals born in October could be a cohort. All right, but the key thing here is that we're taking that single cohort and we're following them from birth until the very last individual dies. And because we're following them, we can then calculate age-specific mortality rates. And this would be the actual age-specific mortality rate. So your mortality rate is the chance of death. What's the probability of dying? All right, and if we follow a cohort, we can calculate the chance of dying for each age, and this is the actual chance of dying because we actually saw them die. We know when they died, and we can know the cause. That's different from a static life table. A static life table takes a sample of our population at a specific time and then breaks them down based on their age category. We then use this distribution to infer our age-specific mortality rate. So we know the largest number of individuals in our population in a specific age should be that earliest age. And then as we age, at every age class, we have a chance of dying, and thus our proportions should decline. Our numbers should decline. We use that information then to make predictions or make estimates of the actual mortality rate. We didn't calculate the actual mortality rate because we didn't follow them. We're just using the age structure to infer it. All right, so cohort life table is actual age-specific mortality. Static life table is inferred mortality rates based on the underlying age structure. So our static life tables can like take a snapshot of the population, or we can go to all individuals. We can say all individuals that died this year, what was their age, and use that to create a static life table. That's what we're actually going to do in lab. So not next week, the week after that. We're going to learn how to create these life tables. And we're going to create static life tables. So how do we do it? How did we do it? Well, we have books. Books in the library. In this case, it's the West Texas Collection had the books. But these are books of, you can say, deaths. All right? Sounds a little bit morbid, but we have books that list usually name of individual and the age in which they died. And it tends to be grouped by, they usually group it by years. So we can go 1951. These are all the individuals that died and their ages. And what you would do is record their ages to come up with the starting age structure. And that's what we did. We actually, I think we broke it down by decades. So for certain decades, we recorded all the individuals that died, their ages, and made the life table, and then you will complete the life table. So as part of the life table calculations in the lab, I do have this, see the lab, know the variable names, know their definitions. You're going to need to know that, and we do have sheets in the lab handouts, and we also have a sheet that I've posted that kind of does the same thing as a review. So why do it? This would be the example of a life table. So you have your age classes. X is our age class, so this is a 0 age class. This is individuals that were born. Individuals that were born at the start of that, or they were alive at the start of this age category, basically. All right, and then you have age class 1, age class 2, age class 3. They do start at 0, and they start at 0 for a specific reason, because that allows us then to calculate some other things. That's part of this utility. All right. So then you have your age class. This is an X, which is the number of individuals that are alive at the start of that age category. So if you are alive at age 0, you were born. You were alive when you were born. And then you can see the numbers drop off. That's expected. We have mortality rates. Then you have DX, which is the number of individuals that died during that age category. And we use both NX and the DX to derive your mortality rates. And the one that we're interested in is the age-specific mortality rate, which we would call survivorship. This is L sub X. L sub X, or survivorship, is probability that you would survive to the next age category, or to that current age category. So if you made it into the population, you survived birth, you have a 100% chance of surviving to birth at that point. All right? So survivorship curves are useful, and we're going to talk about those. Those are coming up. Next thing, we can calculate life expectancies, EX. That's why we need to start at 0. For our age class. And then, using all the information in the table and our birth rates, birth schedules, we'll get to those, we can estimate our population growth rate. So the first thing we're going to do is talk about these survivorship curves, because they're probably one of the main reasons why we create these life tables. All right, so survivorship curve is a graphical representation of age-specific survival. A little bit different than mortality. But if you didn't die, you survived. Hence your survivorship curves. This graph is a plot of the log of survivorship as a function of age. So it's not just LX on the y-axis. It is the log of LX. demographics on the y-axis, and then age on the x-axis. We'll make these in lab. There are three broad patterns of survivorship curves. There's a type 1 curve up here. There's type 2 curves, which are basically straight lines. And there's a type 3 curve that looks like this. All right. So type 1 curves, examples of type 1 curves or organisms that produce type 1 curves would be humans, elephants, anything that usually has a high survival throughout a large part of their life. And then as they reach old age, as they reach the end of their lifespan, mortality rates pick up and their survivorship drops off. So type 1 curves are characterized by low survival in our young. That's our initial drop here. Low survival in the very young, and then high survival, basically our flat line, until we get to old age. And this part, this low survival in the very young is pretty typical. It's just the newborns are susceptible to a lot of things. That's a type 1 curve. Type 2 curve looks something like this, which is a straight line. And birds, good example of these. A lot of birds exhibit these type 2 curves. These type 2 curves come about because you have constant mortality rate. So your chance of dying when you're 2 years old is the same as the chance of dying when you're 3 years old. The same as the chance of dying when you're 4 years old. And that's different from a type 1, where it's like your chance of dying when you're 20 is basically the same as the chance of dying when you're 25 or 30. But then as you get older, once you get up to like 60 and 70, 80, now your chance of dying starts to really increase because we're nearing the end of our lifespan. Not so with the birds. You still have your same probability of death. And then the type 3 curve, you've got low survivorship early in life. Now, again, note the difference. In type 1, I said low survivorship in the very young. In the type 3, I say low survivorship early in life. So during the first parts of our life, or their life, you're going to have high mortality rates, which translate to low mortality. But then, once you get to adulthood, then you basically have high survivorship. Examples of these, we have white oak here and barnacles, invertebrates, great example. Fish, great example. There's a reason why you produce so many offspring. It's because very few of them will actually survive to adulthood. All right, so these are the survivorship curves. And as I was saying, the survivorship curves relate survival. But if you don't survive, then you die. So we can also do mortality curves. So we can also do mortality curves. So what we're going to do is I'm going to draw these curves and show the relationship between survivorship and our mortality. So our type 1 curves, so log mel x, or age, that's our type 1 survivorship curve. See if I can get a different color. Maybe that one. Oh, there's it. It's really red. On this axis, on the second axis, we're going to do mortality, which will be QX. so that's our mortality rate. QX is not on a log scale. QX just relates proportion of individuals that died in that age class. All right, so if we have QX mortality rate, what we're going to see is we've got basically the slope of the line is what is key. When you have a higher slope, you have higher mortality rates. So in a type 3 curve, we have higher mortality rates in the very young, and then we've got low mortality throughout most of their life until we start to reach older age. That's our type 1 curve. Type 2 curves. Type 2. Log of LX, age. We said survivorship is a straight line. How do we get that? I said we get that because our mortality rates are constant all through life. So the proportion that dies in each age group is the same. And that gives us this straight line with a negative slope for the survivorship curve. And then our last one, type 3 curve. Again, log of X, log of LX, our age. Type 3 curves look something like this. And since we have a very high slope, so QX, we got a high slope throughout most of our early life, our QX curve will mimic that. So our QX curve and the survivorship curve for a type 3 type survivorship curves are very similar. Better? Very similar. That's our relationship. If you have high survivorship, you have low mortality. If you have low survivorship, you have high mortality. But our mortality, or the survivorship that we're talking about, based on the slopes of that line. I say this in lab. If you fail to make it on the log scale, all of these survivorship curves will look similar. All right? They'll look similar. They'll look very similar to a type 3 curve. But if you put it on the log scale, that starts to expand the differences and really reveals type 1, a type 2, and a type 3 from each other. So be on that log scale, very important, very important. Questions? Are they related to parental care?
Yes, they are.
You're guessing they are. How?
Like I said, fish have like a lot of offspring, but they're pretty much like if they compare crop. That's pretty much like you spend the first few years of the offspring, travel and everything. That's when you have right.
Survivorship?
Yeah.
Yep. Yeah, so parental care is tied to it. There's quite a few things that are tied to these curves. Parental care is one. So yeah, if you spend a lot of parental care because you might not hit maturity until this age point. So that parental care may be a cause for the high survivorship. Probably the cause for the high survivorship. But in a situation like this, you have almost no parental care. Offspring are on their own. You're going to see a high mortality rates at that point. They're not independent. But these are various life history strategies, and life history strategies could be predicted based on these types of survivorship curves. So yes, we'll come back to that. We'll talk about these life history strategies. And we'll put it in perspective, not just in the demographic characteristics, but also in population growth. Population growth. right? So these are survivorship curves. Comparing curves can reveal very important biological processes. All right, so the key things when we make the comparisons is to look at the slope of the line. All right, are the slopes different or are they parallel? If the slopes are parallel, then we have similar mortality rates in those two populations. If the slopes are different, then we have different mortality rates. And the differences in the slope reveal the magnitude of the mortality rate. So these are two different examples. And we present them because they show differences between habitats and differences between the sexes. So again, these are survivorship curves. It's mislabeled. It should be the log of LX, but I can see that this is on the log scale based on the numbers. So 50, 100, 500, 1,000. This is what a log scale would look like. I just think the label should be log of LX. All right, so autocoileus. What is that? I bet some of you know. Do you know? Anybody? Who's mammalogy? A couple of you are in mammalogy, right? I see somebody else like, yeah, I'm glad I'm not. Shocked. How many of you are hunters? Deer. But this was done, I believe, in California was a study. So you had deer populations in two different habitat types. So you had the chaparral area and the shrubland area. They looked at, so there was differences in density, believe it or not. So they started investigating why. Why would there be differences in density between these two different areas? Well, it seemed like it came down to their survivorship. So in this case, you have higher survivorship, lower mortality in the chaparral regions than in the shrubland regions. All right, so the prediction was actually, well, shrubland should have had a higher densities, but chaparral actually did. And that's reflected by difference in the mortality rate. So note the difference in the slope. The steeper the slope, the higher the mortality rates. You still saw this, death early in life, but then you get higher mortality. So in these two populations, yeah, the chaparral region had higher densities. They also tend to live longer. So it suggested then that perhaps, our view of which area was more productive could be wrong. Maybe the chaparral region was actually a little bit more productive for this deer species. So that's a case where habitat differences produce different survivorship curves. Same species, just two different populations. We would call these two different populations. The other one is capreolus. What's that? Anybody? No one's less likely to know that. Roe deer. So, another deer species. But here, they looked at survivorship curves and they broke it out by sexes, male and female. And what can we tell by this? What can we tell? We've got very different changes. So you've got female survivorship that seemed to be about even slopes at this period as the males. What does that mean? Do we have equal mortality rates? Yeah, it looks like mortality rates are about the same. And then you have difference in slope. And not only a difference in slope, you get a pretty high mortality for the males, less so for the females. And for the most part, the steeper slope persists for the males, which means their lifespan is a few years shorter than the female deer. Why would that difference appear? Why would males have higher mortality rates? Because what? That could be because they're hunted more. Definitely could be 100%. Because what? Competition for? Yeah, for mates. Competition for mates and territory. So yeah, males are fighting, and this is a pretty common occurrence where when you have males that are fighting for territory, fighting for mates, the males tend to exhibit higher mortality rates because you have that risk of getting injured. You have the risk of getting injured. And if you have that risk of getting injured and you do get injured, your mortality rates start to go up. So yeah, the survivorship curve is very important. They allow us to make predictions about life history characteristics, which we'll do. But for us, we're talking about comparing survivorship curves and making inferences as to why some of the curves may have similar mortality rates if they're parallel or different mortality rates if we see that they are not parallel. All right. We're going to be stopping here, so I do have the quiz. Attendance quiz. You're going to see the attendance quiz questions. The answers are all coins. So your answer choices are pennies, nickels, dimes, and quarters. So the answer is 3 that are most common, or have the most in common with each other. Nickel, dimes, and quarters. The only non-copper one. Nickel, dimes, and quarters. And once you're done, once you take that, you are good. We'll come back on Wednesday. We'll do life expectancy. Make sure you review. If you have questions about our test materials, please bring questions. If you have a question, other people might have the same question. We can answer questions on Wednesday. All right. Nickel, dime, quarter. That was our answer. See you Wednesday.